Dear students, our today's topic is the parthenogenesis. When we talk about the parthenogenesis, it refers to the development of an egg without fertilization. You all might have heard about the Greek word parthenos refers to virgin, genesis refers to birth. The individuals formed by the parthenogenesis are called parthenoids. Parthenogenesis, we have two types. We can say the natural parthenogenesis and the artificial. When parthenogenesis occurs spontaneously, it is called natural parthenogenesis. It is a regular natural phenomenon in a few groups of animals. Some animals reproduce exclusively by parthenogenesis. In some other species, parthenogenesis alternates with the sexual reproduction. How we can differentiate in groups the parthenogenesis? In some of the species, you can see the parthenogenesis alternates with the sexual reproduction. Natural parthenogenesis is divided again into two groups, complete and incomplete. When we talk about the complete parthenogenesis in certain animals, parthenogenesis is the only method of reproduction. This type of parthenogenesis is called complete parthenogenesis. Population exhibiting total parthenogenesis consists entirely of females. There are no males in this case. We can give the example, Lacerta is a kind of lizard. Incomplete parthenogenesis in some animals, parthenogenetic reproduction and sexual reproduction occur alternately. This is called incomplete or cyclical parthenogenesis. So we have made here differentiation between complete and incomplete. When I talk about the examples, in golf flies, parthenogenetic reproduction alternates with sexual reproduction. There is one parthenogenetic reproduction and one sexual reproduction per year. In aphids, daphnids, and rotifers, one sexual reproduction in summer after many parthenogenetic reproductions. Natural parthenogenesis is further classified into two types. Again, they are haploid or arenotoky and diploid parthenogenesis that is also called thelytoky. When I talk about the arenotoky, it's a type of parthenogenesis. In this case, a haploid egg develops into a haploid animal without fertilization. It is also called haploid parthenogenesis. It is found in insects, arachnids, and rotifers. In honeybee, parthenogenetic eggs develop into males and fertilized eggs develop into females. And the males are haploid and the females are diploid. Very important point, the males are fertile. During parthenogenesis, the reduction division is omitted. Hence, the spermatozoa contain the haploid number of chromosomes as the parent. The females contain diploid number. The eggs are formed by meiosis, hence they are called haploid. Here you can see the example of parthenogenesis in honeybee. Female is diploid. After meiosis, it divides into two haploid cells. And the single haploid cell due to parthenogenesis develops into male. And the male, due to mitosis, it produces sperm that are also haploid. Sperm after fertilization, they produce female. When we talk about the thelytoky, it's also kind of parthenogenesis. In thelytoky, an unfertilized egg develops into a diploid animal. It is also called diploid parthenogenesis. It is found in crustaceans and insects. The diploid condition is maintained either by reduction division or by the fusion of the one or the polar bodies with the haploid egg. Based on this, thelytoky is classified into two types. I have mentioned here ameiotic and meiotic as well. In ameiotic thelytoky, meiosis is suppressed. The egg is produced by simple mitosis. As a result, the egg is diploid. It generally occurs in crustaceans and insects. When we talk about the meiotic thelytoky, in meiotic, the meiosis occurs during oogenesis and the haploid eggs are produced. The diploid condition is achieved by the doubling of the chromosome. The doubling of the chromosome is called diplosis. Thelytoky by restitution. Here you can see the primary oocyte, first incomplete meiosis, second oocyte polar body. Due to second meiosis, ovum is formed. Here, when we discuss about the diplosis, diplosis occurs in any case of two ways, namely restitution and autofertilization. Restitution, in this process, the first meiotic division is incomplete. The second meiotic division occurs normally. The results in the production of two diploid cells. The smaller cell is called the polar body, while the larger cell is developed into the egg. The diploid egg develops into the embryo without fertilization, for example, Lepidoptera and insects. When we talk about the autofertilization, in this process, meiosis occurs normally during oogenesis and haploid egg is produced. The haploid egg then becomes diploid by fusing with one of the polar bodies. This process is called autofertilization. I will mention in the next diagram. In Artemia saline, it's also called brine shrimp. The process occurs by fusion egg with the second polar body. When we talk about the parthenogenesis in vertebrates, natural parthenogenesis is rare in vertebrates. A few parthenogenetic vertebrates occur in nature. They are as follows. In the case of lizard, Dasypta saxicola armeniaca, there are no males. It produces only diploid egg. The diploid egg develops into diploid females. Before gametogenesis, the chromosomes are duplicated in the sex cells. One more example I have mentioned here, the fish Carassius auratus gibelio reproduces parthenogenetically. Here also there are no males. Here you can see I had mentioned in my previous slide that talking by autofertilization. Then what is the artificial parthenogenesis? Parthenogenesis produced experimentally in the laboratory is called artificial parthenogenesis, as the name itself reveals. This has been demonstrated in annelids, mollusks, echinoderms, amphibians, birds, and mammals. Parthenogenesis may be produced by mechanical or chemical means. When we talk about the chemical means, it is induced by treating the eggs with many chemicals like hypotonic or hypotonic seawater, chloroform, blood serum, fatty acids like lactic acid, butyric acid, they can be used. Fat solvents like ether, acetone, etc., urea, chloride of potassium, calcium, sodium, magnesium, they can also be used. When we discuss about the mechanical means, artificial parthenogenesis is induced by the following mechanical methods: high or low temperature and the ultraviolet rays. Very important point, sometimes it is asked in many examination, the gynogenesis or pseudofertilization. It should be or pseudofertilization. It is a special type of parthenogenesis occurring in a few turbularians, nematodes, and earthworms. In these animals, the egg is stimulated to develop by the penetration of the spermatozoon. What happens after penetration? You can see the sperm disintegrates, very important point, without fusing with the egg nucleus. Thus, the process is said to be gynogenesis or pseudofertilization. Gynogenesis is more common in hermaphroditic animals. What are the significance of parthenogenesis as a means of reproduction, rapid multiplication, persistence of advantageous characters, elimination of disadvantageous characters, elimination of variety, as a means of sex determination, prevention of wastage of energy, polyploidy, fertility, and the inheritance. I hope this topic has been very useful for you all. Thank you. Thank you very much.